All right, let's get started. Hello, my name is Alan Hudson from the Immuno-Oncology Translational Network Data Management Resource Center. I also serve as Chair of Biostatistics and Bioinformatics at Roswell Park Comprehensive Cancer Center. Before we start the seminar, I just have a few bookkeeping items to go over. Questions will be responded to at the end of both presentations towards the top of the hour. Please enter your questions in the Zoom Q&A box. Please do not enter your questions in the chat box. The audience can prioritize the order of the questions by clicking on the thumbs up button. Closed captioning is available. The closed captioning for today's webinar can be accessed by clicking on the live transcript option in the Zoom menu at the bottom of your screen. Instructions are also provided in the chat box. If you're having technical issues, you can make a comment in the Zoom chat box. With that, I would like to turn the proceedings over to Dr. Lilian Kuo, who is a program officer at NCI in the Division of Cancer Biology, and one of the key organizers of the Cancer Moonshot Seminar Series. Lilian?
Thank you, Alan. In support of the next generation of cancer researchers, today's Cancer Moonshot Seminar will feature research from talented junior investigators or early career scientists. These are abstract-driven talks nominated by Cancer Moonshot investigators and selected by NCI staff. We are delighted to feature science from Dr. Nicole Halmai of the Minority Patient-Derived Xenograft Development Trial Centers, or MPDTCs, and Dr. Nick Markham from the Human Tumor Atlas Network, or HTAN. Bringing together these complementary topics and novel technologies to understand gastric and colorectal cancers, we're grateful to have Dr. Ken Lau serve as our moderator. Dr. Lau is Associate Professor of Cell and Developmental Biology at Vanderbilt University. His laboratory utilizes high-content experimental and computational tools on mouse models and human specimens to study signaling between cells. And for the Cancer Moonshot, he serves as PI for the Colon Molecular Atlas Project as part of HTAN. And I'll hand it over to Ken.
Great, thank you for the kind introduction, Lilian. So for our first speaker, we have Dr. Nicole Halmai. So she is currently a postdoctoral fellow in the Carvajal Carmona lab at the UC Davis Genome Center. And Dr. Halmai relatively recently obtained her PhD in 2019. So, started postdoc a couple of years ago. And outside of her scientific research, which was focused on genome editing of human cancer variants, she was also very active in the various diversity and equity initiatives. So for her current research, she is focused on a very important question, dear to my heart actually, is that does racial and ethnical variation affect cancer progression? And I'm very excited to hear the latest and greatest in this aspect.
Great, thank you. So I'll go ahead and share my slides now. How does that look for everybody? Great. All right, thank you so much for the introduction. Good morning and good afternoon to everyone. Thanks for joining and listening to my talk here right before the holidays. So as was mentioned, my name is Nicole Halmai. I recently got married, so that's the name change there. I'm a postdoc in the Carvajal Carmona lab over at the Genome Center at UC Davis. And I want to thank the organizers for allowing me to speak today about our research focused on establishing and characterizing preclinical models to advance our gastric cancer precision health equity. So today, I'm going to talk to you a little bit about gastric cancer. So we know that gastric cancer is the 5th most common cancer type and the 4th leading cause of cancer-related deaths worldwide. And while the overall incidence of gastric cancer has decreased in recent years, the overall prognosis remains poor, where most gastric cancer is diagnosed in advanced stages, and the US 5-year survival rate for metastatic gastric cancer is just 5%. Despite this, we actually know quite a bit about risk factors that are involved in the probability of developing gastric cancer, but also gastric cancer survivorship over a person's lifetime. And these can generally be divided into 4 different categories. You can see here, so environmental, lifestyle, genetic, and societal. So things like workplace exposure, diet, family history, and access to screening have all been reported to have an effect not only on gastric cancer development, but the type of gastric cancer a person can develop. And so thinking about this whole idea of cancer precision medicine, it's critical for us to understand what combinations of cancer risk factors contribute to certain subtypes of gastric cancer that can then be treated most effectively by specific therapeutics. And to complicate things even more, we also know that many of these risk factors are not independent of one another, and their effects are not independent of one another. So perhaps a more accurate depiction of these risk factors is something like this. And in the Carvajal Carmona lab, as was described, we are interested in understanding the effect of these risk factors through the lens of cancer health disparities. And it might not be surprising to you that many of these risk factors here for gastric cancer in general are also factors in cancer health disparity. So most US racial and ethnic minorities are disproportionately affected by gastric cancer compared to non-Hispanic whites. And so what do I mean by this? Well, most US racial and ethnic minorities are 1.5 to 2 times more likely to be diagnosed with and die from gastric cancer. Most minorities are also diagnosed at earlier stages and at more advanced stages of gastric cancer at diagnosis. Interestingly, we also see differences in the actual subtypes of gastric cancer in our minority patient population. For example, we see increased rates of Epstein-Barr virus and Helicobacter pylori positive gastric cancer rates. This is particularly true for our Hispanic Latino population and our Alaskan Native populations. We also observe higher frequencies of the histological diffuse subtype, as well as gastric cancer that originates in the non-cardia region of the stomach. And so why am I telling you this? Why is this important? Well, this might be a familiar figure from a 2014 study coming from TCGA, where they were able to utilize around 300 gastric cancer tumors to generate novel molecular subtypes of gastric cancer. And importantly, these molecular subtypes were associated with specific genetic and epigenetic alterations that are important for therapeutic efficacy. And so again, I just want to point out these 4 subtypes here, which I'll be talking about later on in my presentation, which consist of chromosomal instability, microsatellite instability, EBV positive, which again is one of the risk factors that I mentioned earlier, and the genomically stable subtype. So of course, this is, has been a really critical advancement in our understanding of cancer, and specifically for gastric cancer, towards this idea of precision medicine. But unfortunately, for this study and for many cancer sequencing studies that have been done thus far, they all seem to suffer from a similar caveat, which is a lack of diversity in their patient samples. So this is, again, another not surprising figure where we have depicted the distribution of TCGA cases by self-identified race and ethnicity. And you can see here that less than 1/5 of all cancer cases in the TCGA come from racial and ethnic minorities, and that's combined. It's the same story when we look at things like established cell lines. So in an genetic ancestry admixture analysis of about 1,000 cell lines from the COSMIC database, right, we see the same pattern where the vast majority of samples are coming from individuals of European ancestry. And so in general, overall, most racial and ethnic minority groups in the US specifically are severely underrepresented in the cancer sequencing data sets that basically define novel gene targets and novel therapeutic targets for development, as well as the preclinical models that are used by researchers such as myself in order to model mechanisms of therapeutic resistance and sensitivity. And so this is a really critical gap in our knowledge that's sort of exacerbating a lot of the cancer health disparities, particularly for gastric cancer, that are experienced by racial and ethnic minorities in the US. And so going back to this idea of gastric cancer risk factors, well, in addition to the factors that I described, we know now that race and ethnicity is also another risk factor for gastric cancer development. And like these other factors, it's not independent from one another. We know that race and ethnicity is associated with a number of risk factors that contribute to the type of gastric cancer that we think the racial and ethnic minority populations exhibit. And so this type of goal of including racial and ethnic minorities in future cancer studies to diversify our understanding of the molecular landscapes of these tumors is really the driving force of what we've developed as sort of an infrastructure at UC Davis towards cancer health equity, which is the University of California Patient-Derived Xenograft Development and Trial Centers, or UCAMP. And you heard a little bit about that in the beginning. So this multi-center grant is designed to allow us to generate xenograft and organoid models for 4 different types of cancer: bladder, gastric, lung, and liver, with at least 60% of these samples coming from racial and ethnic minorities. And the idea is that once we generate the models, we can submit these models to a suite of molecular characterizations, which includes whole exome sequencing, RNA sequencing, and DNA methylation, that will allow us to not only molecularly characterize the tumor and the models, but also identify genetic drivers of cancer that might be specific to our racial and ethnic minority patient population. We can also utilize the models in preclinical therapeutic screens, and as well as contribute to the diversification of preclinical models and molecular data for general use. So in that sense, we are depositing all of our xenograft models that we generate in our studies and all of the molecular data to a biobank that will allow general use through our collaboration and involvement with the NCI's PDX-NET consortium. And we're just one of 2 centers in this consortium that are specifically focused on racial and ethnic minorities. So today I'm going to talk a little bit about some of our efforts for gastric cancer specifically. So far we've been able to generate xenograft and organoid lines from 24 gastric cancer samples. And you can see the samples stratified here by self-identified race and ethnicity compared to the gastric cancer cohort from TCGA. And already you can see that while most of the samples, again, are non-Hispanic whites in the TCGA, our cohort conversely is dominated by Hispanic and Latino Americans, which is maybe not surprising with the catchment area of UC Davis here in California. And so thinking about a cohort such as this, which is dominated by minority patients, we wanted to look at the distribution of these molecular subtypes that had been originally identified by TCGA. And so you can see that here at the bottom, while TCGA, again, is dominated by this chromosomal instability subtype, conversely, in our samples, we see over half of the samples are of the genomically stable subtype. And this is important because the genomically stable subtype has been mostly demonstrated to have the fewest number of mutations, which in turn causes it to be one of the more difficult subtypes to treat, just because there are less targets. And so it might be critical moving forward to really understand what types of mutations are associated with the genomically stable subtype in order to better treat our minority gastric cancer patients. And if we step away maybe from predefined categories and just look generally at what the mutation profile is, we also see differences, significant differences from TCGA. So here we just stratified genes by their frequency of mutation, predicted to be deleterious or known activating mutations. And again, we can see distinct differences between our minority patient cohort and TCGA. So while TCGA has the top 2 mutated genes as TP53 and TTN, we have those genes as well, but equally frequently mutated are ARID1A and MUC6. And importantly, we also see a number of genes that we don't see in the TCGA data set. And I want to point out the MUC genes here, where we have MUC6, MUC12, MUC19, and MUC2. So again, these genes encode the mucins, many of which are known to be associated with different types of cancer. And so we see a unique enrichment in our minority population of these MUC genes, and that's something that we want to pursue in future work. Looking at just known cancer driver genes from the OncoKB database, if we look at just tumor suppressor genes and oncogenes that are known to be drivers, again, we're seeing the same thing, right? Less TP53 mutations, which is maybe not surprising because we're seeing fewer of the CIN subtype, which is the main subtype that has TP53 mutations. More ARID1A and APC mutations, and fewer PI3K and ERBB3 and 4 mutations. And so overall, basically what this is showing is that our minority gastric cancer tumors display a distinct pattern of genetic alterations that may be therapeutically relevant. And one of the alteration patterns that we've really honed in on has been these high frequency of alterations in two pathways, the PI3K-AKT pathway and the CDK cell cycle pathway. So if we consider not only single nucleotide variation, but also copy number variation and promoter DNA methylation that causes gene silencing, we see that over a third of all of our cases have alterations in their genetic landscape that's indicative of aberrant signaling in either PI3K or CDK cell cycle. And a third of those actually have aberrant mutations in both. And so this really led us to focus in on these two pathways for our downstream therapeutic screening. So I mentioned that we generate organoid lines for high throughput drug screening, and this is an example of that here with 5 organoid lines that I had described previously. So we use IC50 curves to identify sensitivity and resistance to different drug treatments. This includes a number of chemotherapeutics as well as targeted inhibitors. And importantly, what you can see here with 4 of the organoid lines that I showed previously having these alterations in PI3K and CDK pathways, we see sensitivity to corresponding inhibitors. So for all of the PI3K inhibitors and the CDK inhibitor that we used, we see sensitivity across these cell lines. So not only do they have the alterations that would potentially make them sensitive to the inhibitors, we are actually seeing a response in vitro. And for our top therapeutic, and for our most effective therapeutics, we actually take this a step further and go into our xenograft mouse models. And so again, this is an example of one of those lines that has these alterations in the PI3K and CDK pathways. And you can see that we treated here the xenograft tumor with a number of PI3K inhibitors, an AKT inhibitor, and CDK4/6 inhibitor. We see varying degrees of sensitivity and response to the PI3K inhibitors, as well as the AKT inhibitor that resulted in significant overall survival extension. But really what we saw as the most effective treatment was the CDK4/6 inhibitor here, which you can see in the black triangles, where basically we saw a complete response, almost no tumor growth when the xenograft models were treated with CDK4 and 6. And so with something like this, where we see complete sensitivity, we can actually take that a step further and generate resistance models from these lines. And we can do this by taking these these highly sensitive PDX models and stop treatment, for example, here with the CDK4/6 inhibitor, allowing the tumor to regrow, and then retreating with that same inhibitor, essentially trying to push the tumor cells towards developing new mutations that would confer resistance to these inhibitors. And here you can see an example of that here. We generated a resistance line for this PDX model, where again, when we treat with that highly effective CDK4/6 inhibitor, we're now seeing in purple basically no response. The tumor is able to grow in this environment. However, when we add on the AKT inhibitor that was partially effective in the previous slide that I showed you, we now see inhibition of tumor growth. And so this kind of implies, right, the dynamic between these two pathways and the relationship that they might have in specific therapeutic resistance. We can also take this a step further and really focus on mutation discovery. And so in the last few minutes, I just want to talk about some of the efforts that we're undergoing in the lab in order to functionally model resistance driver mutations. So one of the ways we can do this is by taking the resistance line that I described before and put it through that same molecular characterization in order to identify candidate driver alterations and then model these back in the organoid lines. And one way that we're doing this is by using the CRISPR-Cas9 system. So this is a project that I've led in order to develop a pipeline for us to generate isogenic patient-derived organoid lines that specifically have alterations in specific pathways, leading to aberrant signaling. And so in this example, I'm showing you here that we use the Cas9 system, designing guide RNAs to a shared exon across all isoforms of a major negative regulator of the CDK cell cycle pathway, CDKN2A. We can electroporate Cas9 and guide RNA, as well as a puromycin resistance plasmid into our normal gastric PDO lines. So these are non-cancerous and basically select for cells that are expressing the plasmid based on puromycin resistance. And so after our selection, we can then seed individual organoid lines for expansion and genotyping, which is ultimately validated by protein and mRNA expression for the knockout, in this case for CDKN2A. And ultimately what we get are isogenic lines that are derived from the same parental minority normal gastric organoid line that either is wild type or has aberrant signaling of a specific pathway, in this case, CDK cell cycle. And here's just some of the results from one of our lines that we've been able to generate. Here, the knockout is caused by a single base insertion near the cut site of the guide RNA. And here on the right, you can see bright field images of what those lines actually look like. And incredibly, we see significant differences in not only in morphology, but also in growth rate. So we see the knockout lines growing faster with aberrant CDK cell cycle signaling compared to the wild type, which is something that we would expect. And so again, just going back to this idea of functional validation of cancer driver genes, we can basically take these organoid lines that have this pathway aberrantly signaling in the cells, and then model these candidate resistance mutations on top of that to really get into these mechanistic understandings that are going to define efficacy for therapeutics for patients. And so in summary, I was able to tell you about the UCAMP Trial Center, which is our infrastructure that we've been able to develop to address cancer health disparities for racial and ethnic minorities in the US. We're developing xenograft and organoid models from our racial ethnic minority cancer patients for molecular characterization, preclinical therapeutic screens, and more recently, mechanistic modeling of resistance and sensitivity to specific therapeutics. Our preliminary results show high rates of PI3K, AKT, and CDK cell cycle alterations in our minority gastric cancer patients. And correspondingly, we see response to those inhibitors, both in vitro and in vivo. And again, our ultimate goal is to improve our understanding of genetic drivers and effective therapies for racial and ethnic minority cancer patients that experience gastric cancer health disparities, and ultimately advancing towards cancer health equity. So just quickly, I want to thank everyone in the lab that contributed to this work, all of our UCAMP collaborators, including the two co-Is, Drs. Chen and Pan. Of course, the NCI PDX-NET Consortium for its continued support, as well as our funding sources, and specifically the Diversity Supplement Award that has been able to fund all of my postdoctoral research. So thank you, and I look forward to questions in the panel session.
Great, thank you, Nicole. It was very interesting talk, very translational too. So we'll save the questions and discussion until the end. with the panel discussion. So I'll introduce our next speaker. And it is an honor for me to introduce my esteemed colleague, Dr. Nick Markin, who is a newly minted assistant professor within Vanderbilt University Medical Center, Division of Gastroenterology, Hepatology, and Nutrition. So I got to know Nick a little bit, and I can say that he is the prototypical physician scientist, right? He is a great gastroenterologist and endoscopist, as well as an excellent basic scientist. So his postdoctoral work was done in a joint postdoctoral fellowship with Dr. Bob Coffey and Dr. Borden Lacey at Vanderbilt University Medical Center. And that research has been mostly focused on stem cells, intestinal stem cells, and perhaps cancer stem cells, and their interactions with the microbiome, specifically C. diff. So today, he is going to talk about a project that is associated with our ColonMap project, which is looking at the effect and the effect of C. diff and other microbial components on colorectal cancer initiation and progression.
Thank you very much, Ken. And I also want to give an extra thank you to Bob Coffey, who nominated me for this seminar series. My project is a spin-off, as Ken indicated, of the Vanderbilt ColonMap Molecular Atlas project led by these four investigators, my mentors. And the purpose was really to generate spatial imaging data across normal, early, and late precancer progression, and to define the microbiome and specifically the colorectal cancer associated biofilm impact using single-cell RNA sequencing, multiplex immunofluorescence, and fluorescence in situ hybridization, or FISH, to analyze the biofilm, and as well as exome sequencing. And this is a unique project because we consider the microbial component as a part of the tumor microenvironment. So colonic bacteria have long been associated with colorectal cancer. This is the unfortunate view of an adenocarcinoma through a colonoscopy. And if we could look closer, perhaps we'd see millions and millions of bacteria that are directly touching the epithelium. And if we understood better the interactions between these bacteria and the colonic epithelium, then maybe we'd have better targeted therapies, maybe even probiotic immunotherapies, or even vaccines. At the very least, we hope to gain better insight into surveillance colonoscopy intervals. Now, specific bacteria have been associated with tumorigenesis and inducing colorectal cancer. From this review by Wendy Garrett in Science a couple of years ago, she highlights Fusobacterium nucleatum, Enterotoxigenic Bacteroides fragilis, or ETBF, as well as PKS positive E. coli that produces colibactin. And work done by one of my mentors, Cindy Sears, has shown in the past couple of years that in a mouse model of familial adenomatous polyposis, or FAP, both of these organisms, PKS positive E. coli and ETBF, are required for the induction of colorectal cancer, not PKS positive E. coli or ETBF by themselves, and not the combination of those organisms lacking pathogenicity loci. And these data are quantified here on the right. So we think about bacteria contributing to tumorigenesis both as a direct effect on the epithelium, but shown here also as an indirect through propagation of a pro-tumorigenic immune response. And some bacteria, as you can see here, will be protective, but others, and again, I'll highlight ETBF, induce an immune response that participates and promotes the tumorigenic process. So we also think about bacteria and changes in the microbiome as dysbiosis. And in many, many different disease states, we have seen associations with reduced diversity of the microbiome or altered community structure of the microbiome, or in the cases that I mentioned before, pro-tumorigenic species. And those are all compositional changes. We can also think of bacterial dysbiosis as structural changes. And here I'll introduce this concept of biofilms. So in the normal colon, the bacteria reside in the outer mucus layer in the lumen and are separated from the gut epithelium by a sterile inner mucus layer. When bacteria invade into that inner mucus layer and directly touch the epithelial cells, they form an invasive biofilm. You can see in this EM from one of Dr. Sears's publications in PNAS that you have these rod-shaped bacteria, potentially E. coli, pseudocolored in purple, with intermixing with strands of mucus pseudocolored in yellow. We've shown this in mouse models as well. This is a publication that we had with Ken Lau's group using a method to preserve the mucus during colon fixation. And that allowed us to simultaneously do immunofluorescence to look at epithelial proteins shown here in green and red, as well as fish in gray to look at bacteria. And you see the inner mucus layer separates them nicely. Now, when that gets invaded, as is shown here in Julia Drew's paper in MPJ biofilms and microbiomes, the fish staining is in magenta and shows bacteria directly overlying the deep purple epithelial cells of a human colorectal cancer. You can also use species-specific probes in fish, as Julia has shown here in this Fusobacterium bloom, intermixing with other species inside and around a human colorectal cancer. To better understand how human biofilms interact with the epithelium and ask the question of whether or not they are tumorigenic, Sarah Tomkovich and others, including Julia Drews, who is with Cindy Sears's lab and is now an assistant professor at Johns Hopkins, combined biofilm positive tumor samples and created a mixed biofilm slurry, gavaged them into APC-Min mice and looked at the colon 10 to 15 weeks later. Mice that were gavaged with the combined biofilm positive bacterial slurries induced tumorigenesis, whereas mice gavaged with biofilm negative samples did not. The next thing that we wanted to do was tease out what are the components of the biofilm that are tumorigenic. So we separated out the bacterial slurry into individual patient slurries. So instead of combining, we separated them and found that there was one biofilm positive slurry that was particularly pro-tumorigenic compared to some of the non-tumorigenic ones. Looking more carefully at the components of those biofilms through 16S sequencing, we were able to see the specific bacterial species. And this really wasn't that striking. There weren't any obvious telltale differences in the pro-tumorigenic versus non-tumorigenic that jumped out right away. It is worth saying that when we compared the sequencing of the slurries to the sequencing of the mouse feces, and these were in germ-free mice, so the feces are a representation of what the slurry is. The slurry often, or the feces often did not contain the same proportion of bacteria in the slurry. And that's known that this is a mouse model and the mice don't take everything that's given to them. But we did decide to move forward with an experiment comparing one pro-tumorigenic slurry with one non-tumorigenic slurry. And the pro-tumorigenic slurry that I'm going to discuss came from an ascending colon cancer that was KRAS wild type, and the non-tumorigenic came from a sequel stage 3 cancer that was KRAS mutant. So what we did is we took the non-tumorigenic slurry and gavaged it once into germ-free APC-Min mice. We took the pro-tumorigenic slurry and gavaged it into a separate group of APC-Min germ-free mice. The non-tumorigenic slurry led to no tumors. The pro-tumorigenic slurry led to tumors. When we look more carefully at the biofilms, we could see that they did form in the distal portion of the colon in the mouse, particularly at about 7 days. And we could score this with a bacterial score. And you'll find that a mucus score that looks at the thickness of the mucus showed the inverse. After 2 weeks, the biofilm presence began to decrease as the thickness of the mucus increased. And you can see that represented here in the H&Es, where you have the luminal contents directly opposing the epithelium here at 7 days. The inner mucus layer then forms at 2 weeks, but the damage is already done. We've seen microadenomas form at that early 2-week time point. We also look carefully at the epithelium. We notice that there's increased beta-catenin staining as well as increased crypt depth length in mice given the protumorigenic slurry compared to non-tumorigenic. We also found increased numbers of dysplastic foci and microadenomas in the protumorigenic slurry inoculated mice at 2 weeks. These dysplastic foci can also be seen on H&E and are evident by their altered crypt structure, increased cell density, and increased nuclear cytoplasmic ratio. As time goes on, starting at 2 weeks, we see an increase in the number of microadenomas and adenomas in mice given the protumorigenic slurry. Using single-cell RNA sequencing and looking specifically at epithelial clusters from the 2-week time point, we see segregation within those clusters depending on inoculation. So here are the different cell types, colonocytes that are secretory and absorptive, stem and progenitor cells, goblet cells. And you see that there is a nice spread between samples that were from mice given protumorigenic versus non-tumorigenic in blue and orange respectively. In the non-tumorigenic slurry inoculated mice, we saw more differentiated colonocytes and fewer stem cells, as one might predict, and in a fairly equal number of goblet cells. When we look closely at the goblet cells, we found an increase in Reg4 expression among protumorigenic slurry inoculated mouse goblet cells. And that's interesting because Reg4 has been observed as a marker for deep crypt secretory cells. These cells are supportive of the LGR5 positive stem cell population in the colon, and they're also a product of increased Wnt pathway activation. We looked more carefully at Wnt pathway activation by looking at gene expression within progenitor cells of specific Wnt target genes and found that they were increased in the protumorigenic slurry shown across the top. We also looked at differentiated colonocytes and found that they had an increase in the expression of genes related to production of reactive oxygen species. Below, I'm showing a table of our gene set enrichment analysis, looking at different specific cell types in different analyses, and found some interesting things, including enrichment for some pathways that are immunoregulatory, increase in genes related to PD-1 signaling, as well as increase in type 2 interferon gamma signaling, among others. So we next move forward to dissect out the components of those two biofilm positive slurries, the non-tumorigenic and the protumorigenic, as I showed before. Once we have put these into mice, then with the help of Julia Drews, as I mentioned before, my colleague at Johns Hopkins, she had done an advanced anaerobic culturomic experiment to isolate 30 bacterial cultures or 30 isolates that were derived from either the slurry or the mouse feces, and then sequenced them in order to discover their species, and found that Clostridium difficile was among the bacteria that were present in the protumorigenic slurry. And that was interesting. So we decided to then take those 30 isolates and include C. diff or remove C. diff and put them in 2 separate groups of mice. And lo and behold, this isolates plus C. diff given to the mice induced distal colon tumorigenesis, whereas the isolates without C. diff did not induce tumorigenesis. So this consortium of 30 isolates was able to reproduce the tumorigenic phenotype of the protumorigenic slurry. And this is looking at colon tumor numbers after about 10 weeks in the germ-free APC-min mice. Now, the 30 isolates did not include any known tumorigenic bacteria, no PCAS-positive E. coli, ETBF, or Fusobacterium. The protumorigenic consortium turns out it requires toxigenic C. difficile to induce tumorigenesis. So without the C. difficile isolate, the protumorigenic consortium did not induce tumors. With the C. difficile isolate, it does. And with a separate clinically derived C. difficile isolate from a separate biofilm also induces tumorigenesis. And beyond that, if you take the C. diff isolate and add it to the non-tumorigenic slurry, it then develops distal colon tumors in those mice. And this is what they look like. Here's a sham inoculated mouse with a normal colon stained with methylene blue. The isolates minus C. diff also doesn't induce tumors. And here are the images of the gross tumors in mice that had been given isolates with 2 of the different clinically derived C. difficile isolates. It turns out the tumorigenic phenotype appears to require C. difficile toxin B. So C. difficile, as a part of its infectious pathogenesis, produces 3 different toxins. The 2 that really drive the inflammation and epithelial injury are TCD-B and TCD-A. Using the protumorigenic isolates and then adding 1 of 4 isogenic strains of C. difficile from the M7404 strain, we find that strains lacking TCD-B do not induce distal colon tumorigenesis, and strains that have TCD-B do induce tumorigenesis. With the help of Reese Nipple, a former graduate student at Vanderbilt, and Eric Scar's lab, who's now a postdoctoral fellow with Cindy Sears' group, we're able to quantify the stool toxin titers, having a maximum toxin titer at about 4 weeks, and then correlated that with the presence of colon tumors at 12 weeks. And the mice that had more toxin at 4 weeks ended up having more tumors at 12 weeks. We also have shown that colonization of specific pathogen-free mice with toxigenic C. difficile could reproduce the tumorigenic phenotype. So it wasn't anything special about the germ-free mice or necessarily about the other isolates, but C. difficile, toxigenic C. difficile, either an ATCC strain or the toxigenic clinical strain, could induce distal tumors in APC-min mice that were in conventional specific pathogen-free housing. And our understanding of this suggests that perhaps colonization may be more important for tumorigenesis than infection. When we look carefully at the biofilms in these mice at the 2-week time point, we find that C. difficile induces crypt-invasive biofilms in mice that were gavaged with the protumorigenic isolates. So without C. difficile, you see the intermucus layer present in these fish experiments, with a few exceptions here with the arrowheads. But when you add C. difficile, there's this influx of bacteria, all bacteria shown in magenta, specifically the C. diff bacteria in yellow, getting deep into the crypts where they normally wouldn't be. And here I'll blow up this picture a little bit more so you can see some of the C. difficile. They're not the most abundant organism, and there's certainly other organisms that are crawling in there as well. Looking at the immune response using high dimensional flow, we found that C. difficile and protumorigenic isolates induce a Th2, Treg, Th17, and gamma delta T cell infiltration, and that some of those populations have an elevated PD-1 expression consistent with the protumorigenic immune response. Here we see that Th17 cells have high PD-1 expression and IL-17 expression, and gamma delta T17 cells also have that increased PD-1 expression. So to summarize, I'll tell you that colon cancer associated biofilms and C. difficile induce tumorigenesis. We found that colonic bacteria can form biofilms that have tumorigenic potential. We found that C. difficile is found in colorectal cancer associated biofilms. And we found that toxigenic C. difficile specifically is sufficient for inducing tumorigenesis in this model. One of the most exciting future directions I have for this project is to use spatial transcriptomics to understand how biofilms interact with colonic neoplasia and reveal potentially how epithelium autonomous versus immune-mediated tumorigenic mechanisms are at play. This is just a screenshot from the experiment that we finished, and we just got the data back last week, and so I don't have an full analysis done yet, but we're looking forward to seeing how these different stromal and epithelial regions will reveal transcriptomic insights into what's going on in these microadenomas versus the opposing normal tissue from these mice given protumorigenic isolates plus C. diff. There's a ton of people to thank who participated in this project, particularly my mentors, Bob Coffey, Ken Lau, Martha Shrubsall, and Cindy Sears, as well as Borden Lacey at Vanderbilt, among many others. Thank you.
Thank you, Nick. Nice talk. So I would just like to remind you, if you have a question, please enter it in the Q&A box and I will ask them for you. So I think we are now at the discussion panel stage. So I'll just start off with asking a couple of questions. So this one is for Dr. Hamai, and I'm just going to ask it, in my own words, kind of thing, right?
Sure.
There are these like fancy algorithms, right, where you can predict the etiology from the mutations. Like, oh, if it's a smoking mutational signature, if it's a, APOBAC mutational signature, et cetera, et cetera. Have you tried applying some of those to your mutational signatures in your models?
Yeah, that's a great question. So we haven't done that yet, but that's definitely in the works. I think, especially for a lot of the racial and ethnic minority groups where we only have a couple patients so far, I think we definitely need to get, a bigger picture, a bigger sample size before we can really look at those. But yeah, I mean, definitely.
And do you collect the lifestyle, risk factor type of information? Because the idea is maybe you can correlate what you find in that mutational study to the actual, risk factor, right, involved.
Right. Yeah. So we do try to collect as much as we can. I think so far it's really focused on, any possible comorbidities or previous diagnosis, in the stomach, like any inflammation or things like that. And then also if there's any infections, right? So focusing on EBV and H. pylori. We know that H. pylori, for example, has a very specific signature of inflammation in gastric cancer. So yeah, we do have some, but it would be great to have more, definitely.
Nick, this question is for you. And it's a question that I'm interested in. So your C. diff strains that causes tumors, and there are other ones also, right? Are these the same bugs that are common in infection?
Yeah, so we've done, with the help of Seth Walk, we've done some strain typing of the clinical isolates, and they are the same ribotypes as those found in infections that have been plaguing hospitals. I mean, C. difficile infection is the most common nosocomial disease in the United States. And these toxigenic strains are very similar, if not the same ribotype as infectious C. diff.
Cool. So another question for Nicole. So, your models, most of them, right? Correct me if I'm wrong, is you're getting the tumor cells from the patient and either do it as a PDX or do it as a PDO organoid, right? So in that situation, maybe some of the environmental factors are missing. So how do you think about that? Immune cells, microbiome, different nutritional, environment, et cetera, et cetera, et cetera.
Yeah, you bet. Yeah, so obviously, I think there's a lot of risk factors that I think might be difficult to capture just with our xenograft and organoid models. But I do think there are ways that we are working towards that. So I think first and foremost, really, we're just trying to create a platform that generates the data, right? Right now, we don't even have any data whatsoever for a lot of these racial and ethnic minority populations. So I think just having that sequencing data first is going to be that first step in order to generate these hypothesis-driven downstream experiments. But I think with the organoid models, that's really opening a lot of doors, I think, for treating the organoids with specific carcinogens or different infections even, right? Things like H. pylori and looking at the effects of that. Of course, it is just epithelial cells, but I think also organoids are being used in these multi cell type co-cultures now with immune cells. So I think there's a lot of possibilities, especially with the organoid models that we have for manipulation and understanding the underlying mechanisms of these factors.
So Nick, these two questions related, and I think it's quite interesting. What do you think about the biofilm status and different species found in a mismatch repair deficient background, like in terms of MSI high cancers and so forth? And also related to that, what about the distribution of these crypt invasive biofilms? Is it uniform along the colon, or is it distributed in a specific regional specific fashion?
Yeah, so there is work that's recently been done looking at the biofilm components of tumors from different, different types of tumors, and that includes different types of adenomas, precancerous polyps. I'm not as familiar specifically, and I'm pretty sure that they had not found C. difficile in those experiments. Within the distribution, we clearly see more biofilm in human on the right-sided lesions as opposed to left-sided lesions, although they are there in the left-sided. It's nearly, biofilm positivity is nearly universal in the right-sided. However, in the mouse models, it's really just limited to the distal colon, and it's temporary. So it's been a little bit harder to get a handle. The microbiota, human microbiota, even in the dysbiotic setting, is very specific to humans. And it does make some sense that it would be different and not taken up 100% by mice. It may be that mice aren't the best model to study the components of the biofilms, but it does seem to be a decent model to study the tumorigenic effect.
Nicole, this is a challenging question, I think, for you. So at Howard University Hospital, which targets African Americans, they don't see the level of gastric cancer that you guys reported. Any clue to that? Because actually what they have seen is that it's actually less than white populations.
Sure. Yeah. I think this question is really important, I think, because it looks into elements of our understanding of racial and ethnic categories that I think are going to be really critical moving forward in understanding cancer health disparities. So I think one example I can give is for American Indians, which I actually know really well. So, overall, there's a certain incidence and mortality rates for American Indians. But when we separate them by the regions where they live, there's a huge variation. And we see this also among the Asian American population, right? Different subpopulations of Asian Americans, which is a hugely diverse category, right, of individuals. They also have extremely varying distributions of incidence and mortality. So I do think the generalization and categorization into these racial and ethnic groups is something that we might need to consider about, what kind of caveats we're incorporating by doing these large groups. And maybe in the case of the area around Howard, there might be specific other, non-genetic ancestry associated, environmental or lifestyle factors that might be contributing to that incidence difference.
Yeah, I mean, there's a question here that suggests this, right? Are you capturing the different strains of H. pylori in your population? Maybe different, different regional locations. It may be. Are you?
Yeah. Absolutely, yeah. So I don't have that data now, but we're trying to look into that. Because of course there have been reports of different levels of frequency of different strains in different populations. So, and that kind of associates, I think, also with things like diet and lifestyle choices. So, yeah, all really great questions that I think are going to be important moving forward.
So Nick, this question, I saw this question here, but it's gone now. Maybe you answered it. But have you looked into microbes inside cells?
Yes. So in other studies, individual intracellular bacteria have been seen inside tumor cells, but C. difficile is not an intracellular organism. That's not a part of its pathogenesis. So we haven't focused on that. There probably is an opportunity there for us to look using some of the universal bacterial probes in fish to look for intracellular organisms, but it hasn't been a focus of our work, but I know others have seen it.
So Nicole, this is my question actually for me. So you see mutation in MUC2, right? Mm-hmm. MUC2 is an intestinal mucin. It's not a gastric mucin. So I'm just wondering for specimens associated with that mutation, do you see metaplasia? Do you see stomach becoming intestine, right? And have you sort of, and does that carry into your models? I think that's a major question.
Right. Yeah. So, I wasn't able to, I think, go in depth into it just because of time. But I think these are the questions that we're looking into right now. So we're doing the histological, assessment of the original tumors from the patients and trying to understand that better. But it does seem, for example, that this pattern enrichment in the MUX is also carried over into that genomically stable subtype, which is often a diffuse subtype also. So I do think there is definitely a correlation with histological patterns for sure, which is not surprising, right? We know that these are important for a lot of elements of cancer progression and development and invasion. So I don't have the answer yet, but we're looking into it.
But I mean, actually, as a follow-up to that, this is what my interest, right? But so you talk about different histological differences. Do you consider that when you're building your models? Because you can imagine that they need different conditions to
Yes.
maintain those histological conditions.
Yeah, absolutely. And you know what, I think in gastric cancer, we've had a big problem with that because the diffuse subtype is actually really difficult to generate organoid models from, right? They just usually don't take very well. And so we are trying to understand, what kinds of culture conditions are going to be able to help us generate these models, which are missing really, right? We have mostly the intestinal subtype. And obviously the diffuse subtype is going to be important for our racial and ethnic minorities because it seems to be enriched genomically stable diffuse subtypes. And so we need the models to understand it. But yeah, it's definitely something to consider moving forward.
So Nick, have you guys looked into pathways that lead to toxin production and secretion? Can that be, like, for example, can that be used as a therapeutic target? Since, it's toxin dependent, right? The effect.
Yeah, that's a great question. The toxins are large proteins, and the genes and pathogenicity islands that encode these toxins are well understood. That's an active area of research within the infectious disease community targeting treatment for infectious pathogenesis. We do know a bit about that, and there haven't been small molecules directed directly at the toxins because the toxin receptor interactions are still not fully understood. There are neutralizing antibodies that have been made and are FDA approved to prevent C. difficile recurrence. Now, whether or not that's going to be a helpful therapy in colon cancer, we're way too early to know about that. We're still just now understanding the relationship between this organism and cancer.
Ken, probably one more question.
All right, one more question for both of you, I guess. This is the question that was previously typed. So what is the status of microbial dysbiosis in underrepresented populations? Oh, is that a food for thought type of question?
Yeah, that's a great question. I mean, there's a whole field of microbiome research dedicated to understanding what is different and normal and not normal in different populations. I mean, with the sensitivity of these assays, we can tell who lives in what household. So within specific populations, there's still a lot of diversity. And to answer the question, what is normal versus what is dysbiotic is not even all that clear.
Nicole, you have any?
Yeah, I think I would just echo that. I think we've really sort of, at least my understanding, we've kind of focused in on specific, dysregulation patterns, specific infections, specific species. But again, I think looking at it as a whole and understanding, everything that is the microbiome, we're still working on it, I think. Yeah.
With that, I think we've come to the top of the hour. Thank you, Drs. Markham and Halmai, for a great set of talks and a great discussion. Thank you, Dr. Lau, for your moderation. We look forward to our next talk on January 27th, generation of multi-epitope specific T cells for cancer therapy by Dr. Rule from Dana-Farber. And thanks again, everyone.